do want to let you know that we'll be taking questions throughout the broadcast. You can use the questions tab that's on your control panel to send us questions, and if we have time left over, we will get those answers as best we can, and I can try to take some offline otherwise. And with that, I will go ahead and introduce today's speaker. A. Wesley Burks is the Curnen Distinguished Professor and Chair of the Department of Pediatrics at the North Carolina Children's Hospital in Chapel Hill, North Carolina. Dr. Burks has been published in many distinguished journals and has authored or co-authored numerous articles, chapters, and abstracts. He is a member of the NIH Hypersensitivity, Autoimmune, and Immune-Mediated Diseases Study section, and is past president of the American Academy of Allergy, Asthma, and Immunology. And with that, I'll pass the mic over to Dr. Burks.
So, Mike, thank you. It's nice to talk with you today. What I'd like to do for the next few minutes is to start off with a little bit of background that many of you know about food allergy, and then begin to talk about some of the treatment options that are being developed or under development. First, we know the prevalence of food allergy, the number of children, almost 6 million school-aged children in the U.S. There's been almost a 20% increase in the last 15 years. Why more food allergy is a fascinating question. Many people think about it, obviously, and many people are writing about it. Some of the writings of Dr. Graham Rook from the United Kingdom, I think, have been the best written about this in the last few years, and I would commend those to you. Specifically, if we talk about peanut allergy, we know that in the industrialized societies, about 1% of kids have this disease now. It's the most common cause of anaphylaxis in children that will present to an emergency department. It's the most common cause also of life-ending food anaphylaxis. The standard of care for food allergy right now is the diagnosis appropriately made and then avoidance of that food. That's still how I practice in clinical medicine. We'll talk about some of the options today that are under development and where we hope things will be in the next several years. Certainly the use of antihistamines, self-injectable epinephrine at the right time, at the right place are appropriate, and again, we'll talk about a little bit about that. But most importantly, there's not a proactive therapy available right now, and that's what we'll talk about, things that are under development. So in the clinical studies that I'll reference in the next few minutes, there are two terms that are often used. The first one is desensitization. And when I use that term, what I mean is that while you're on a study, while you're on a treatment, the amount of food that you'll tolerate on a food challenge is different. So prior to the study, you may react to a third of a peanut. While you're on the study, you can tolerate the ingestion of 15 peanuts. But that's only while on treatment, and that's the desensitization effect. Tolerance, meaning you started treatment, you see changes clinically, and then you stop the treatment, and then at some point off treatment, you can still tolerate the same amount that you did before. Interestingly, in the field of food allergy, there's not a good definition of what tolerance might be. An article that was published about 18 months ago in the New England Journal of Medicine about egg oral immunotherapy that we'll talk about later talked about the development of sustained unresponsiveness. Another word, again, what we might all describe as tolerance, meaning you are on a treatment, you have an effect, you stop the treatment, and that effect is long-lasting. So the other things I'll try to define as we get started, the different phases of the Food and Drug Administration, the FDA trials that we have, from phase 0, which is really the preclinical development, early safety type studies, and then phases 1, 2, and 3, which are actually the clinical studies themselves, and then phase 4 after a product is released on the market. So I'll refer to each of those in some way as we talk about each of the different types of studies. And the second terms to try to define are the design of some of the studies. The first, randomization, or randomized, meaning as you enter a study, by prior arrangement, there is a randomization schedule, a table, and it's done many different ways that if you're the first person entered, you'll be randomized to a treatment or a placebo, or one of several treatments or placebo. Blind, meaning as you enter the study, you don't know, the person entering the study doesn't know which treatment they are on because the treatments are masked in such a way that you can't tell. And then placebo-controlled, meaning on a study, if you're trying to study treatment A, there's treatment A, which is intervention, and then there's the placebo, which is an inert substance that should not have any immunologic or clinical effect, as we're talking about. What we do know in clinical studies is that there's about a 25 to 30% placebo rate of change, not immunologically, but clinically in any of the studies. And so we'll talk about how that kind of factors in today. So this is a cartoon from an article from the Journal of Allergy and Clinical Immunology from 2011 that on the top half talks about the clinical trials that are in process, and it's pretty much the same now, except for some modifications. And then the bottom half are the preclinical studies. On the upper left, that column are the allergen-specific studies. So if you're allergic to milk, you'll get milk treatment. On the right are allergen nonspecific. If you're allergic to milk and egg and peanut, you'll get this type of treatment. So one's allergen-specific and one's not allergen-specific, and we'll talk about studies from both of them. Also, the paradigm that I'll try to describe as we go through each of the studies in the types of immunotherapy that we're talking about typically happens that as you enter the study, the dose will be really very low so that it is safe and it doesn't cause any adverse allergic reactions. Immunologically, you have to build up to have an effect, but you start out with a dose that would be judged to be safe based on previous studies. And then over time, that dose is increased, and it may be daily, it may be every other day, it may be once a week, it may be bi-weekly. There's lots of different paradigms, but it's similar in each of the studies that we'll talk about. You start out with a low dose and build up to a higher dose, and that higher dose where I've shown is called the maintenance dosing, and that maintenance dosing would be carried on for a period of a phase of several weeks to months to years, again, depending on the study. And at the end of that time, that desensitization term we talked about earlier, you do a food challenge while they're on the treatment. So that's the desensitization part. And then at some point, then you discontinue the treatment, you continue to avoid the food, and then that tolerance food challenge or that sustained unresponsiveness. So that time period here can be days, weeks, months, years, just again, depending on the study and the definitions. So how long, that's the definition that we really don't know. How long do you need to be off before you can truly is tolerant? So now we're going to come back to the question that we started with to talk about the first series of studies on extensively heated milk or egg in the diet. And these are studies that have come primarily out of Mount Sinai. Led by Dr. Simpson and others in his group. Their thought was that the current treatment right now is that if you're allergic to egg, idea that, well, maybe what happens as you outgrow the food allergy, which we know will happen the majority of the time for milk and egg, that you put the baked or heated product into the diet, and that will help accelerate the loss of that food allergy. So this study that referenced here is in milk allergic children. The mean age was a little bit less than 7 years, and they were challenged sequentially to products that had baked milk in them, so baked muffin, waffle, and then uncooked milk, to find out how many of the children, how many of these 100 children could tolerate a heated or baked product. And a rough definition of baked heated for milk or egg is about 350 to 15 or 20 minutes, and it varies depending on the study and the particular person talking about it. But you can see here in the challenges that about 75% of those children that were milk allergic could tolerate the heated milk product as they were given the challenge. So in that 75% of children, what was seen in that study and others like that is that it was associated as that food was put into the diet. So these are milk allergic children that tolerate a heated milk challenge but couldn't tolerate uncooked milk. Their milk-specific IgE, their milk prick skin test, their basophils, which are white blood cells that react to milk, and their lymphocytes, the T regulatory cells, which are types of lymphocytes, again, that help control the immune response, they all changed with the introduction of the baked product into the diet compared to those that weren't on that. So after a couple of years of treatment, it seemed to accelerate the development of loss of that allergy this particular study, which is milk allergy, but others that they and others have done in egg allergy, similar findings. So the questions that are under development now are an effective dose, what degree of heating, or can that change over time, and then the role of the food matrix. Again, these are exciting studies, though, that have gone on, and I think for many people that take care of a number of children with food allergy, it has changed their clinical practice in allowing or encouraging this to happen in milk and egg allergic children. So the next studies, as we come back to our overall big picture cartoon, are to talk about oral immune therapy. And I'll contrast this with sublingual immune therapy. First, to talk about oral immune therapy, this is if you're allergic to peanuts, we'll take a peanut powder that has all the peanut proteins in it, mix it in another food like applesauce, mix it well, and then take the whole product, the powder plus the applesauce, and eat that. So that's the oral dose. Sublingual, as you can see on this slide, is a liquid extract with a dropper, and those drops are placed underneath the tongue. So there are differences in oral. You can get more quantity of the food, whatever it might be, the route of delivery, and then digestion and how that may affect it. So oral immune therapy is the powder of the food mixed with other foods typically. The sublingual is a liquid concentrate of that particular food put underneath the tongue, held for several minutes, and then swallowed. So there have been multiple studies that have been conducted, worldwide on oral immunotherapy, and I'll talk about it, a couple of them. The same paradigm, though, in all the studies that have been published, low dose to high dose, a period of time on maintenance therapy, and then a challenge on therapy, and then sometimes a challenge off of therapy. The thing that you have to try to pay attention to as you go through this is that the outcome from any of the studies is very dependent on the dose that the individual's being given, how long they're on the maintenance, and then how long they're off the treatment before that tolerance or that sustained unresponsiveness challenge is done. The outcome will be different at 6 months, at 12 months, at 3 years, and 5 years. And that's the part of trying to understand the studies. So the first one we'll talk about is the Consortium of Food Allergy Research, or COFAR, an egg oral immunotherapy study, and then a peanut oral immunotherapy study. So the COFAR egg oral immunotherapy study was published, as I said, about 18 months ago in the New England Journal of Medicine. These were 55 egg allergic subjects greater than 5 years of age. In the treatment, 40 of the subjects, the children, were on egg oral immunotherapy, and 15 were on placebo. It was done at 5 centers: Mount Sinai, Johns Hopkins, here in North Carolina, Arkansas Children's, and National Jewish. Those were the centers involved in the study. So the investigators and families didn't know as they entered whether they were one of the 40 on egg or the 15 on placebo, because it was blinded, and the treatment continued through 48 weeks. So with that as a background, what I'll show you now is at the end of 10 months of treatment, there was a challenge with food being done. So this is a desensitization challenge. So each individual that entered the study had a daily treatment. They came back every several weeks to several months to the treatment center. And then at the end of 10 months, they were given a food challenge, 5 grams of egg. And you can see in the placebo group that none of those passed that 5-gram challenge. And in the egg oral immunotherapy group, that 55% of those that were on the egg oral immune therapy could pass that challenge. So now we're going to continue the treatment for another 12 months. So the first one was 10 months, now another 12 months. And at the end of 22 months, a 10-gram challenge was done. So they're still on treatment. None of the placebo could tolerate, but now 75% of those that were in the treatment group could tolerate that 10-gram challenge. So it's gone to 55% to 75% in 1 year. So now we're going to, that's the desensitization effect. So now we're going to talk about the tolerance. So at 2 years of treatment, 75% could tolerate it, but now you stop the treatment for 4 weeks, and you can see now it's down to less than 30%. So from 75% to 27.5%, that many people didn't tolerate taking off the treatment. It changed the amount of food that they would tolerate. So now we're going to watch as this study continues another 12 months, and now we're out to 3 years, and again do a 10-gram challenge. Now the statistics are that a little bit less than a third of them could tolerate it at 2 years, and now at 3 years, you're up to almost 50%. So you can see the differences. The longer the treatment with the same dose allows more kids to come off the treatment and be able to tolerate the food. So now we're going to talk about the next study. This is a peanut oral immune therapy study that was published a couple of years ago. There were 25 subjects in this. 16 were on active treatment and 9 were on placebo. And it's the same paradigm that we followed earlier, from a low dose to a high dose, and then they continued that maintenance with either the peanut treatment or peanut placebo. And at the end of the year, you can see that on the y-axis here, that's 5,000 milligrams roughly, that everyone that was on the peanut oral immune therapy could tolerate that amount. So 5,000 milligrams of peanut, and you can put that in terms of about 15 to 20 peanuts. And then those that were on placebo tolerated less than a third of a peanut. So this is while on the treatment. So if you're on the treatment, you would tolerate 15 or 20 peanuts. If you were not on treatment, you would tolerate less than a third of a peanut. So if we look at those subjects longer term, so 19 of them completed the whole protocol, which ranged from a little bit less than 3 years to greater than 5 years. So we wanted to find out what would happen after 3 or 4 or 5 years of treatment. If you stop the treatment, what happened? So 11 of those 19 can now eat peanut in their diet without symptoms after stopping it. But these are really small numbers of subjects that were actually on the study. So it's encouraging, but again, cautious in the interpretation of it. If we look at the immune response, so what happens with oral immune therapy? So there are different types of white blood cells that we all have that are important parts of our immune system. And regulatory T cells that I have outlined here, then on the y-axis is the scientific nomenclature to try to identify that regulatory T cell. And on the x-axis is how long you've been on the study. So at time 0, then they had very few T regulatory cells that were specific for peanut. As you then went to 12 months on the study, you can see the numbers of T regulatory cells for peanut went up. But by 24 months, here you can see the numbers of them went down. They may function. We anticipated the T regulatory cells would go up and stay up over time. It's an phenomenon and how that might work. If we look at interleukin-5, it's another one of the proteins in the body, a cytokine that we produce. Interleukin-5 is important in the allergic immune response, and if we're going to dampen the allergic immune response, what you would hope would happen, and that's seen here, is that interleukin-5, specifically for when that person's cells come in contact with peanut, changes over time. And similarly, if you look at interleukin-13, which this graph shows, it's again involved in the allergic immune response. And those of us that are allergic produce lots of IL-5 and lots of IL-13, these two that I've shown, in response to the allergic response. And you can see IL-13 goes down similarly with the treatment. And then the last part of the laboratory studies I'll show is the interleukin-10 to interleukin-13 ratio. So IL-10 is another one of the proteins or cytokines that are produced that are not anti-allergic, but we produce more of it if we're less likely to be allergic. And you can see that ratio changes over time because there's less of the interleukin-10 and more of the IL-13 if you're on the study compared to placebo. So now we'll leave oral immune therapy and some of the, again, promising results, but not knowing what happens long-term, particularly after you stop treatment. We'll talk about sublingual immune therapy, as I introduced a few minutes ago. So again, there are fewer studies on sublingual immune therapy worldwide. Talk about a couple of them. One from COFAR, and then one from here in North Carolina that was done. So the first one on the COFAR peanut sublingual immune therapy study, these were 40 subjects, and they were adolescents and young adults, and they were equal numbers on peanut sublingual immune therapy or placebo. And the difference here is that compared to the other studies that we talk about, these are adolescents and young adults compared to younger children. And the other studies that I discussed were primarily children that started studies between ages 4 and 7. So you can see now Same paradigm of low dose to high dose, and then treated for a period of 44 weeks initially, that compared to the beginning where they could tolerate 30 milligrams, which is a little bit less than a tenth of a peanut. By 44 weeks on the study, most of these individuals could tolerate about 600 or 700 milligrams of peanut, which is between 1.5 and 2 peanuts. And if you continue through week 68, they could tolerate about 1,000 milligrams, which is, again, the equivalent of about 3 peanuts. So less than a tenth of a peanut prior to the study and 3 peanuts at the end of 68 weeks of treatment. That amount obviously is different, but it's really different than the oral immunotherapy, which we talked about in the range of 15 or 20 peanuts a few minutes ago. So now I'll talk about a study that was done here in North Carolina on younger children. So these were children that were peanut allergic between ages 1 and 11, and their peanut-specific IgE was greater than 7, and they had a history of having a reaction to peanuts previously. So they were randomized, and that randomization was blind to the subject and family entering, as well as the doctors that took care of them. So they either got the actual treatment, SLIT with peanut in it, or sublingual immunotherapy with placebo in it. And then they underwent a 5-gram challenge after about 12 months on therapy. So you can see here that those on treatment on the left could tolerate a mean of about 1,700 milligrams of peanut. Those on placebo tolerated less than about 100 milligrams. But the striking thing you'll notice here in sublingual is that in comparison to oral, when everyone was on treatment tolerated that higher amount, sublingual, there's a large variability in the amount of food they'll tolerate after 12 months of therapy. And on the right, what you'll notice are the skin prick tests. So these are the sizes of the swelling or the wheal that are measured. Those on placebo, it didn't change during therapy, while those on sublingual immunotherapy, their skin test size actually went down during that year. So some of the knowledge gaps that we'll talk about, and the reason oral and sublingual therapy are really not ready for clinical practice right now, is that what do we know? We understand that desensitization actually begins within a few hours, days, months of treatment. So that threshold, if you can tolerate the treatment, will actually go up. But the allergic side effects, particularly with oral immunotherapy, are primarily gastrointestinal at the beginning. That somewhere between 10 and 20% of children entering a study with oral immunotherapy will not be able to tolerate it because they have significant gastrointestinal effects. It causes abdominal pain significantly on a daily basis. It can cause vomiting. Most of the time when the study is stopped, the treatment is taken out of the child's diet. By 4 to 6 weeks later, the child's symptoms are back to baseline, so it doesn't appear to be long-lasting. But again, we don't know that. We also understand that children with viral infections, that if they develop fever while on the daily dosing, they can tolerate, while they're on the treatment, 5 peanuts, and then they develop fever that day and they take the same treatment, they'll have systemic anaphylaxis. And 2 days later, without fever, they take the same dose and tolerate it without symptoms. So the viral infection and its effect on the immune system is pretty significant and really precludes some of the things that we're talking about. And then for about 20% of children on the studies, exercise in combination with the treatment will cause them to have symptoms. The child takes the daily dosing, they exercise, and then have symptoms, whereas if they didn't exercise and just took the daily dosing, they no longer had symptoms. And then lastly, the side effects. Off of therapy, there's been a recent report from Johns Hopkins on a milk oral immunotherapy study about those children that were actually on the study, followed several years, contacted, their symptoms for milk allergy were actually worse. As we begin any type of investigation, what everyone that participates in the study, whether it's the people that design the study or the families and the subjects that enter into the study, is the hope would be to understand why the treatment either works or it doesn't work. And that's as we describe the mechanism, a possible understanding of how the study might work. And these types of cells, the mast cells, the basophils, the B cells, and the T cells are cells that are in our immune system that we know with allergic disease they react a certain way. And with other types of allergen immunotherapy, what will happen is that those cells will change in their reactivity for whatever type of treatment you're receiving during that study or long term. So there are lots of studies that will look at these types of cells to see if the study worked, maybe why it worked, so the mechanism of it, or if the study didn't work, maybe why it didn't work, so we can design another study that will improve on that. And then lastly, the tolerance, the term we talked about before, that's really not been shown in long-term blinded studies for any of the types of therapy that we're talking about today. For someone being treated and then stopping the therapy and a long-lasting effect that will allow them to tolerate the food. So now we're going to come back to our cartoon and talk about another type of treatment that is being developed, epicutaneous immune therapy. This is a study that is being done primarily by a company in France, DBV. Some of the studies that initially done in Western Europe and now in the United States and other places. And the paradigm for this is that, as shown here, that there is a patch, and it's about the size of a U.S. quarter, and around on the adhesive side, in between that, then as it's shown here, then the peanut itself, or milk, whatever the type of study, has been electrostatically put into the patch. So then you can take the patch itself with the adhesive and put on to the skin, and then as the patch is applied over time, whether it's a day or 2 days, then that food itself absorbs into the skin and gets presented to the immune system. So this is done on a regular basis, again, daily or every other day. Sometimes it's the same dose, sometimes it's the same updosing scheme that we talked about earlier. So I'm going to talk about a couple of studies. The first one is a phase 2 study, which we talked earlier about phases 0 through 4. A phase 2 study is looking a little bit about the safety of a product or the intervention, and then really about how effective it is. So it's a small study. And that's all of phase 2 studies. And in this one, there were 16 children that had cow's milk allergy. 9 were on treatment, 7 on placebo. They had a food challenge as they entered the study. And then after 3 months, they had a food challenge to see if there were differences in their treatment. Then they had a second milk challenge after 3 months of treatment longer. So then what we've seen is other studies with the same type of therapy, with the epicutaneous immune therapy, a study, the Child study in France, the VIPE study that is in both Western Europe and the U.S., and then COFAR will be involved in one of these types of studies. So what I hope all of us learn out of these is how the product might be safe first, and then how effective it might be in both the desensitization as well as long-term tolerance. The early studies, as shown in the milk slide I showed, show that some desensitization effect actually happens. So now I want to talk about the types of therapy that are allergen non-specific. So if you're allergic to multiple foods, it might be one treatment that would help for all of them. So the first one we'll talk about is anti-IgE, which is a monoclonal antibody, so an antibody made. The brand name for this is omalizumab, which is being used now for allergic asthma and other diseases, and it's been used in a couple of trials for peanut allergy. The first one was done in the early 2000s and was published in 2003. That was an anti-IgE antibody that was kind of the parent, so to speak, of the present antibody. And so individuals that were peanut allergic entered into the study, and those that were on the higher doses of the anti-IgE, that desensitization effect happened. So before the study, they could tolerate a certain amount, while on the treatment, they could tolerate a much higher amount. But about 25% or a quarter of the subjects didn't respond to the treatment. So then you go on several years, and something was published a couple of years ago now. There was a second major study, this time on omalizumab, in 26 subjects, and the study overall was stopped prematurely because of safety issues that occurred because of reactions during the challenge, not because of the product itself. So again, what was seen there is that those that were on treatment, that desensitization effect happened. So those on treatment versus those on placebo could tolerate a higher amount on the food challenge itself. I think what we'll see now in the use of anti-IgE is likely, rather than being used by itself as a type of therapy, is that it would be used as an adjunct to other types of immune therapy. So if you're peanut allergic and you wanted to get peanut oral immune therapy, you would get anti-IgE for a period of a few weeks to months, then start the peanut oral immunotherapy, continue both of them for a while, and then stop the anti-IgE and continue with the peanut oral immunotherapy. One study I reference here with milk, there's been a recent one published in the last few weeks with peanut. Both showed that the time to go from the first day to the maintenance dose could be done more quickly and a little bit safer. The overall prevalence, the side effects aren't that much different, but it allowed the dosing to go much more quickly. So we'll see what happens with the use of anti-IgE either alone or, more importantly, with as an adjunct of other type of treatment. Again, going back to our cartoon to talk about the allergen non-specific types of treatment, the next one we'll come to is Chinese herbal therapy. These are studies done primarily out of Mount Sinai by Dr. Xiumin Li and Dr. Hugh Sampson and others. So what was known is that there were demonstrable benefits of Chinese herbal therapy that were utilized in a mouse model of peanut allergy. So the investigators made a mouse allergic to peanuts and then used this type of combination of herbs to treat the mice, and those that were on treatment versus those on placebo had a very different effect. So an herbal formulation was developed to give to humans, the FAHF, so the food allergy herbal formula, the second generation of it, was developed as a formulation of 9 herbs that to be used in clinical trials. So there was an initial phase 1 study, so that's really about safety of a product. And the subjects in that received either the tablets or placebo 3 times a week, or 3 times a day for a week, and the treatment was tolerated well with a few minor GI effects. So then a second phase 2 clinical trial was done. That study the results have not been released yet, but has been completed. So it was in adolescents and adults with allergy to peanuts, tree nuts, sesame, fish, or shellfish. So exciting studies. It'll be interesting to see the results. The novel thing about this is that if you're allergic to multiple foods, it would be one treatment rather than individual treatments if you're allergic to peanut and milk and egg. So what I'd like to come back to is that the first thing that I would say is that the treatment for food allergy right now is an appropriate diagnosis and then elimination of that food. If you're allergic to milk or egg and tolerated product, then maybe that in your diet. But otherwise, it's appropriate diagnosis of those foods. And the story I'd like to tell that I've used before is that if the diagnosis of food allergy is a puzzle and it has 100 pieces, about 99 of those pieces are a clinical history. What type of food did you eat or ingest? How long afterwards did you have symptoms? What type of symptoms were they? Each time you got the food, were the symptoms reproducible? If you thought you're milk allergic and you didn't have symptoms after yogurt, then it's probably not milk because those are the same proteins. So putting all that together as a puzzle, then laboratory studies are that last piece, and then you have the diagnosis and you can help someone either avoid the food or often now put that food back into their diet. What I've tried to show from the studies that we've talked about with oral, sublingual, to some degree epicutaneous, anti-IgE, Chinese herbal therapy, that while on treatment, there is a clinical desensitization effect, that the threshold goes up. It's higher in oral versus sublingual. It's higher in other studies versus some. But there's a clinical effect. If you're on therapy, the threshold will go up. What we don't know is, can you continue that treatment long enough so that there's long-lasting effect of that treatment? You can treat someone, stop the treatment, and that benefit continues. And that's really what the biggest thing that's unknown. Other types of allergy immune therapy studies primarily have been done for insect sting or venom immunotherapy, and then for allergic bites, either grass or weed pollen immunotherapy primarily. The benefit of those has been lasting, that you do have some long-lasting benefit. The difference in food allergy and particularly those studies on allergic rhinitis is that for food allergy, we're either allergic or we're not allergic. You can eat the food or you can't eat the food. If you can eat the food, you eat it without symptoms. If you can't eat the food, then you eat it and you have allergic symptoms. There's not a gradation of it. In allergic rhinitis, you can have fewer symptoms and feel much better and do really quite well. You still have some minor symptoms. that's really not allowed for treatment. So the main question is, can we induce long-lasting tolerance in the types of treatments that we're talking about? I think the discover the whole community is, what's the goal of therapy? Is it sensitization to raise the threshold, or is it a treatment that will produce long-lasting tolerance between the treatment and the person can tolerate the food for the rest of life? Certainly, as you talk to different groups of people that are involved in the subject's family, the study coordinators, investigators, we each come at the question with a little bit different goal in mind. And it's important as we discuss each of the studies that we think about what our goal personally is, and then what the goal really is. So I'll go ahead and stop right now, and I'd particularly like to thank the groups of people that some of the studies that I've shown today that I've worked with, particularly Brian Vickery and Mike Poulos here in North Carolina, Stacy Jones in Arkansas, and Hugh Sampson at Mount Sinai, and obviously many other people that have worked on studies like this. Each of the types of studies that each of you may be familiar with and have talked about will have a number of people involved in this because of the numbers that take to run a clinical study. So I'll go ahead and stop now, and we'll look forward to having discussion and many questions with you. So thank you very much.
So thank you, Dr. Burks. So I'll go ahead and read a couple of questions. We've gotten lots of questions in. We'll try to cover as many as we can. The first question is, can you just describe what might exclude someone from a study? The severity of the previous reactions, if they have asthma, if they have IgE levels that are extremely high, just what might exclude someone from a study.
Okay, so for most of the studies that people are doing now, the original studies excluded several types of people. Most of them right now are children that would have had a pre-reaction, had a severe systemic anaphylactic reaction with hypertension, so their blood pressure would have gone. The exclusion criteria short of that, then if you've had a serious allergic reaction, you could still enter the study. It's just if you've had hypertension, you wouldn't be in it. The other two maintenance would be children that have severe asthma and severe atopic dermatitis, so that their disease is not controlled well. Prior to it would be difficult to immune study without that. And that would be for the oral and sublingual immunotherapy studies, those children that have had a history of eosinophilic esophagitis or gastroenteritis, then they would be excluded. And I can say for most of the studies that we've had the opportunity to be involved with, it's been quite rare to find someone that would be excluded from the study.
So we have a question from someone whose son is undergoing a SLIT, and the difference between IgG4 test results compared to IgE test results, how they interpret those.
Okay. Allergen-specific IgG4. Back up a little bit and talk about IgE is the allergic protein that you have to make if you're allergic to something. Because IgE does not mean you're allergic, though. So as a diagnostic test, the absence of IgE is a good indication you're not allergic. It's not 100%, but close. If you have IgE, it means that you may be allergic. If you have a clinical reaction, you obviously are. So in the immunotherapy studies, as we talked about, you'll follow the skin prick test size, the allergen-specific IgE, the allergen-specific IgG4, as well as the mast cells and other lymphocytes that we talked about. So in an immunotherapy study, the allergen-specific IgE typically goes up for the first 6 to 12 months of the study and then begins to go back down. By the end of year 2, it typically is lower than it was at the beginning of the study. And then depending on the length of the study, at year 3 to year 5, it could be much lower. The IgG4 goes up, starts to go up in about 3 to 6 months, and will continue to go up until about 2 years on the study, and then begins to flatten out after that. Both of them are associated with other types of successful allergen immune therapy, like the venom immunotherapy and the immunotherapy for allergic rhinitis. Their observations, they aren't tied into if the therapy is effective or not, at least from what we understand right now. They're byproducts of the immune therapy studies.
Great. Any thought behind the Chinese herbal therapy and what mechanism might cause it to actually work?
So the question would be about the mechanism of the Chinese herbal therapy and how it might work. And as someone not doing the studies, I'll speak very naively in this, and those that are doing the studies at Mount Sinai and other places could have a much better, more cogent thought about what that might be. So the effects to work generically, so if Chinese herbal medicine will work on multiple foods, it wouldn't necessarily be working on IgE or IgG4 because that's allergen specific, that the effects on the cells themselves, the mast cells and the basophils, and their ability to respond to milk and egg, whatever it might be, or peanuts or fish, whatever the person's allergic to, it would dampen that response. So the ingestion of that herbal formulation would change your mast cells and basophils either temporarily while you're on the study or permanently if it works long term, so that those cells would no longer respond to IgE and then the ingested milk or egg or whatever food you might be allergic to.
We've got a couple questions, just about basic questions about how the clinical the trials work. One question was, can you explain why placebos are important? The question was if OIT is clearly working, and why is placebos needed? If people are worried about doing the trials anyway, they want the real deal. So,
so it's a really good question about any type of clinical study design, why there's a placebo. So as you think about the development of a new investigation, a product, we talked about phases 0 through 4 of the FDA approval process. Phase 0, the beginnings of it. Phase 1, safety primarily. Phase 2, small efficacy. And Phase 3, larger numbers of efficacy. And Phase 4, after a product is approved, to continue to follow to see if there are unanticipated side effects from the smaller studies. So to be able to look at the efficacy of a product, we know that there is a placebo effect in almost any type of study. And it's on the clinical effects, not necessarily on the immunologic effects. So in a typical study for allergic runny nose, allergic rhinitis, there is about a 30% placebo effect. So individuals on placebo, of 100 of them, about 30 of them perceive that their symptoms are better, even though they received not the treatment product, they received water or saline or whatever that might be. And that 30% placebo effect in clinical studies, how do you perceive your illness? The question being asked, other ways to look at that, is always there. So if a treatment didn't have a placebo and there was only a 10% improvement, that's actually less than placebo. You couldn't tell if it was really effective or not effective. So that's the major reason. The second reason is to look at the side effects. We know children that are allergic have other diseases like atopic dermatitis, asthma. They actually get other foods that they're allergic to. And how this treatment might affect us, we wouldn't know unless we had a placebo. And then long term, we need to understand what the normal natural history would be of peanut allergy. And if we didn't have a group on placebo to compare to those on treatment, we really wouldn't understand what might happen without that type of treatment. So even as difficult as a placebo is to do in children, that the long term and even really the short term benefits are huge by including that, so it doesn't encourage us to continue to study treatments that really are not better than placebo.
Great. Another question just about the trial phases. Typically, how long does it take to say get from phase 2 of a trial to say phase 4 or phase 5?
So the question is the timing of the different studies and how long it takes. And if we look at like the immunotherapy that we're talking about, we talk about other types of drugs, medications that any of us have read about, some might be taking. That the interval between phase 2 and then approval of a product after phase 3, phase 4 is post-marketing as the treatment's being used. So how long phase 2 and how long phase 3 takes really depends on the study drug itself and then the outcome of the first studies. If something in phase 1 is really safe, no side effects at all, and the effect is really large, then the time amount for phase 2 and phase 3, if it holds up, will be pretty short. If there are a number of side effects in phase 1 and early phase 2, and you have to study a larger number of people, then phase 2 and phase 3 will be longer. So the safety of the product, the efficacy of the product really guide how long that treatment is. And that's a major discussion with the regulatory agencies as they approve or don't approve products that are being submitted to them, is the safety and the efficacy and how they balance those.
Great. We've had many questions about are we any closer to understanding what the cause of food allergies are, why we're seeing an increase. And in fact, someone said, well, if we can make a mouse allergic to peanuts, doesn't that tell us how people become allergic to peanuts in the first place.
So there are lots of good questions that all of us have about why there's more allergic disease now. So in the context of the big picture, then we know that there is more allergic disease now than there were 50 years ago. We know there's more autoimmune disease now than there was 50 years ago. Certainly what we all are interested in food allergy, that change in prevalence in the last 15 to 20 years seems to have outpaced even other allergic diseases that there's an increase in. But we'll just talk about generally increase in allergic diseases. So other pieces of information are helpful in the discussion. If you look at individuals moving from non-industrialized societies, Southeast Asia, Africa, to industrialized societies, Western Europe, the U.S., that same person in that generation will have more allergic disease in the industrialized societies. There is actually more allergic disease in the non-industrialized societies against Southeast Asia, Africa, other places than there was 20 years ago also. It's just not the same change has happened. So what's different about our lifestyle in the industrialized societies, the amount and types of the foods that we're eating just in general, the air that we breathe, particularly diesel particle exhaust, our microbiome, which are the bacteria that line, the millions of bacteria that line our gastrointestinal tract, are different. And that's just 3 or 4 of the things that are really different in the industrialized societies. So if you try to create a mouse model that make it allergic, if you do one or a couple of those things I just described, then it will help make the mouse allergic. What's hard to tease out is it one of those? Is it a combination of those? Is it all of them? And it's an experiment that can't be done because all those things have happened. We can't change the air that we breathe. We can't change necessarily the foods that we're eating and how they're before we eat them. And then our microbiome is just part of the society that we live in now. And that's not a condemnation of like our society at all, so please don't misunderstand that. It's just an observation. We know that happens. And so how to stop that, then I think the observations that we have here and then the observations that we have in the non-industrialized societies can help us in a big picture from a societal standpoint try to stop or slow down some of that progression.
Can you talk a little bit more about the long-term effects that are seen after some OIT therapies? So, for example, development of EE or just the return of the allergenicity?
Okay, so the long-term effects of oral immune therapy, as we talked about, the most important allergic side effect that really precludes its development right now is the development of abdominal symptoms. And it happens almost always, if it's going to, within the first few days to weeks of therapy. So it's not like someone's been on a treatment for 2 years and then develops abdominal symptoms typically. It's usually literally within the first few days to weeks. And as they take the dosing, they begin to have abdominal symptoms, stomach hurting, pain, not point tenderness, not one particular place, just diffuse across the abdomen. That happens within a few hours of dosing, not right away. So it's not like you take the dose at 8 and your stomach hurts at 10 after 8. You may take the dose at 8 and your stomach begins to hurt at noon and last all afternoon. And then you might have vomiting that afternoon. And as you continue the dose, it continues to happen. And as you stop the treatment, then most individuals will no longer have symptoms within 4 to 6 weeks after stopping treatment. So you stop the treatment at the end of September. By November 1st, middle of November, most of them no longer have symptoms. There have been very few that have continued beyond that time frame. So withdrawal of the treatment allows them to go back to the baseline. So the other part of the question is what happens after a year or 2 or 3 years of treatment, but then the treatment is stopped because the study stopped, then go forward another year or 2. And there are conflicting studies that are there. Some studies would suggest that the people that were on the study a few years off, in fact, their symptoms may be, in fact, or what they're perceiving their symptoms may be, in fact, worse because they're experiencing more allergic reactions. And then other studies suggest that there's not really any change post-study compared to pre-study for those that entered into a study for 3 years. So there's conflicting evidence there, so we'll have to see as more studies come out what the long-term effects are if a study is stopped.
Great. Can you talk a little bit, any research being done between allergies and autoimmune diseases like Crohn's?
So there are, as I said earlier, if we think about allergic diseases and autoimmune diseases like Crohn's, other types of colitis, then there's an increase in both of the types of diseases, allergic and autoimmune diseases that I alluded to earlier. The actual tie-in for children that have food allergy to have more autoimmune disease, it's not more than the general population, and the opposite being those that have some type of inflammatory bowel disease like Crohn's disease, do they have more food allergy? And they don't. It's different parts, but related parts of the immune system that are involved in both, but they do not appear to have a direct effect on either in either direction.
Several questions, if you could expand any further on the OIT plus Xolair study.
Okay. So Xolair is the molecule that binds IgE, so it's an anti-IgE. So if you think about IgE as a stick figure with the arms sticking up in the air, that's like a Y, and that anti-IgE binds to the base of that molecule. And when it does that, it prevents it from sitting on a cell, and the IgE has to sit on a cell and then be exposed to in this case, the food that you're allergic to. So you have to have the food attaching to IgE to attach to the cell, and then the cell breaks open and then the mediators cause the clinical symptoms. So if you think about anti-IgE binding to that IgE, it won't sit on the cell. So if that doesn't happen, then in theory, if you bind all the IgE, then you shouldn't have allergic symptoms. So the thought was that you could use anti-IgE, the first studies I've talked about by itself, and then could you have a long-term benefit? We saw that on the treatment, then the amount of food you tolerated generally went up, but not in everyone. Not everyone could you bind enough IgE to prevent symptoms. So then the thought was, can you use anti-IgE and use oral immune therapy safer or get there more quickly? And so the two studies that have been published and then some others that are there that have seen preliminary results about, if you go to time from point A at the beginning to point B, which is the maintenance therapy, that time from A to B can be reduced on the anti-IgE, and there are fewer somewhat side effects to it. If you look at the whole study from point A to point B, then the anti-IgE is stopped at that point typically, and then the oral immune therapy continues. So it's allowed you to get from point A to point B faster, maybe a little bit safer. Then you have to think about are the side effects of anti-IgE enough of a benefit to help offset the fewer side effects of the oral immune therapy, and right now it appears that they may be.
You mentioned some of the SLIT studies are looking at young adults as well. Are there any plans for or current studies looking specifically at adults only?
So talk about sublingual immune therapy and the different types of studies. Most of the studies right now are being done in children and young adolescents. There are studies that are being planned for adults, different parts of the world, depending on where a person might live, but primarily related to peanut, tree nut, milk, egg.
Okay. One more question, this is the softball. Are you accepting new patients?
So I think every site that does clinical studies in food allergy would like to have more capacity. That the numbers of people that would like to participate in studies, which I think overall is a positive thing. And so I'll back up just a little bit to answer that. I think as you talk to a family about a study, then you have two thoughts as you come into it. You try to talk about all the information about the safety of a study, the intended effects. You try to predict the unintended side effects. And for each family, it's a time commitment. And so for some families, it's the right answer to do the study. In others, it's not the right answer because of different factors. Both of them are right answers. So it's very individual. There's not a right answer for it. But any site that's doing studies definitely would like more people. I think the rate limiting factor for all of us is just the number of personnel that can do the studies through the grants that everybody is using to try to run the studies. So we'd like to do more subjects to come through. It's just trying to make sure that we have enough personnel and money to do the studies.
Okay, great. I think that's it. Great. Dr. Burks, thank you so much for participating in this today. And thanks everyone for joining us. Just give you a quick preview of our next webinar, which will be on Wednesday, January the It'll be addressing food allergies in restaurants, training and legislation. We just had one in November regarding kind of the customer side of things when you go into a restaurant. But this will be the plans that we have along with the National Restaurant Association for training restaurants, and also potential legislation that we'll be seeking coming up in 2014. So David Crownover, who's the product manager for ServSafe, will be joining us. Member registration will open this Friday, December the 13th, and then open registration for everyone will begin on Monday, December the 23rd. So again, thank you for helping make this first half of a year for our webinar series successful. That's going to conclude today's broadcast. We will have the recorded webinar up on our site in about a week in case you missed any piece of it or you want to share it with others. And again, thank you, Dr. Burks, and that will conclude the webinar.